Good evening, everyone. My name is Alexander Kim, and I'm a chemical engineering major. I've had the privilege of working with Dr. Bao in the Chemical and Biological Engineering Department on a project developing iron-oxide nanoparticles for biomedical applications. So to begin, what are iron-oxide nanoparticles? Well, they are essentially nanoscale particles with an iron oxide core typically coated with a polymer or other molecule or molecular structure as seen in this diagram over here below. And these iron oxide nanoparticles have a variety of biomedical applications. The ones we'll primarily be focusing on are the formation of nanoghosts and antibody conjugates. As you can see on the bottom left is the way that we form nanoghosts. So essentially what you would do is suspend your nanoparticles in a solution of cell membrane fragments. And these cell membrane fragments will actually naturally reassemble a cell membrane around the nanoparticle, resembling the original cell membrane with all of its membrane receptors and components intact. On the bottom right, you can see antibody conjugation, where antibodies will actually interact with the polymer coating of a nanoparticle to actually form the conjugate. In addition to these, iron oxide nanoparticles can also be used as MRI markers and for the purposes of drug and protein screening, as well as delivery of drugs. So the big idea behind my project is that cancer cells, their membranes have a variety of receptors, and these membrane receptor components are conferred from the cancer cells to extracellular vesicles, which are essentially the signaling and delivery vehicles of the body between these cells that have their own membrane receptors and membrane receptor components. In addition to this, we have our nanoghosts, which use membrane receptors from membrane receptor components and membrane fragments from these original cancer cells to mimic them. And this naturally leads to our research questions and objectives over here. First, what is the membrane receptor overlap confer from cells to EVs from nanoghosts? And two, can nanoghosts be used to accurately identify ligand-receptor interactions in cancer cells and EVs? And you might be wondering why we're asking these questions in the first place. Well, it's simple. If we can use nanoghosts as tools to accurately determine what compounds cancer cell membrane receptors interact with, we can more quickly and effectively determine what potential drugs might be useful as real-world treatment options for cancer. And this leads to our initial objective of actually synthesizing iron oxide nanoparticles of various polymers to create biological platforms that we can use to answer these questions. So over here are the various polymer coatings that I've worked with, particularly polyacrylic acid, PAA, and polyethylene glycol, PEG, are especially interesting because of their ability to form iron oxide nanoparticles that can be used to form nanoghosts. In addition to this, dopamine and iron oxide nanoparticles can be used for antibody conjugation, and chitosan oligosaccharide iron oxide nanoparticles can be used for cell protein analysis. So with our PAA, PEG, and chitosan iron oxide nanoparticles, we see a general reaction procedure as shown in the diagram to the left. And we have a variety of variable parameters that we can optimize, such as the molar ratio of polymer to iron, the mass of polymer iron, heating time, etc. And our approach is to essentially identify optimal conditions to form iron oxide nanoparticles of the size range and average that we want, and make sure they're uniform so that they can properly form the biological platforms we want. And we do this by iteratively testing different protocols while isolating for individual variable conditions. Over here, we see a TEM, or transmission electron microscopy images, where essentially TEM, with the TEM microscope, you are shining a beam of electrons down a thin grid sample. And this returns a high-resolution image on the nanoscale, where we see the actual iron oxide cores that are reflecting these electrons. And you can see, as seen here, our iron oxide nanoparticles are generally individually around 200 to 300 nanometers, which is the size we want, and are generally uniform. With DLS, or dynamic light scattering characterization, we essentially shine a variety of lights onto a liquid sample containing our nanoparticles. And this returns a quantitative size distribution, as you can see here. And now you'll notice that with chitosan, there's a slight shift to the right in the average peak. And in polyethylene glycol, we see 2 peaks. The reason for this is something called clustering, where nanoparticles will essentially interact with each other and tend to get closer to each other to the point where the dynamic light scattering machine interprets these linked nanoparticles as one structure, hence returning a larger size. However, this can easily be fixed with something called sonication, where we essentially suspend our iron-oxide nanoparticles in a water bath with an electric current running to separate them so that this doesn't interfere with any of our experiments in the future. With dopamine iron-oxide nanoparticle formation, we had to take a slightly different approach because dopamine iron-oxide nanoparticles become very unstable upon exposure to the much higher heat in the previous reaction formula, where you're essentially putting them into a hydrothermal reactor of 200 degrees Celsius, which is why we actually had to start off with a different type of nanoparticle, sodium citrate iron oxide nanoparticles, and then coat this with a secondary layer of dopamine polymer. We then isolated the dopamine nanoparticles through the last steps below by bubbling oxygen out of solution to prevent any dopamine-oxygen reactions, and then freeze dry it for an actual solid sample that we can use for FTIR, or Fourier transformer infrared spectroscopy characterization, where with FTIR, you're basically shining light of various wavelengths and returning the absorbance of that light at each wavelength. And what you get is essentially a molecular fingerprint of every important structural component present in your chemical sample. And over here, we see absorbance peaks indicating structural components that show successful formation of our dopamine iron oxide nanoparticles. Some of the peaks are slightly obscured by suspected water and carbon dioxide impurities. However, the existence of other peaks still confirm successful dopamine nanoparticle formation. In the future, I would like to conjugate various antibodies to our dopamine nanoparticles to form antibody conjugate dopamine nanoparticles to study the transmembrane receptor overlap between extracellular vesicles and cells of cancer cells specifically. In the first few steps, we can see the process of actually conjugating our antibodies to our dopamine nanoparticles. And in the last step, you can see how we would extract EVs and cells of expressing interesting membrane receptor components. In summary, iron oxide nanoparticles can be used to create effective biological platforms, and the type of polymer coating used can pass down special properties that we can utilize to answer questions of interest, especially to find transmembrane receptor overlap conferred from cancer cells to EVs and nanoghosts. These are my references, and thank you for listening. Any questions?
Okay, so I'm personally curious about, you're trying to study these membrane receptor interactions. So why don't you use actual cancer cells instead of nanoghosts?
Well, that's a great question. Actual cancer cells that we analyze in the lab are alive, and actually maintaining and upkeeping them is very both expensive in terms of time and material. Whereas nanoghosts are inorganic and not alive. So it's much easier to analyze interactions using an inorganic, non-alive platform rather than an actual living thing that you have to keep up with.
Is there any other either metal or molecule other than iron oxide that could potentially be used?
Yes, absolutely. There's actually precedent regarding the creation of dopamine nanoparticles with gold instead of iron. However, this has a different set of properties and lacks the same superparamagnetism that we can use to actually extract EVs and cells of interest by using magnetic extraction.
Why were you interested in dopamine as a nanoparticle coating for the creation of antibody conjugates?
That's a great question. Dopamine actually has a special functional group called a catechol group, which upon interaction with a more basic environment transforms into a very reactive quinone group that links very well with antibodies, making it a very attractive polymer coating for antibody conjugation.